Hello everyone, welcome to We Learn and welcome back to the class Molecular Basis of Inheritances. In last class of Molecular Basis of Inheritances, we talked about the structure of DNA. In this class, we will talk about the structure of DNA which was given by Watson and Crick. So if we talk about the Watson and Crick structure, this is what we call three-dimensional structure of DNA. So the structure given by Watson and Crick is also known as three-dimensional structure of DNA. Watson and Crick derived this structure from the basic information provided by Wilkin and Franklin and Chargaff's rule. So the basic information here is the same. What they did is they manipulated this information and basically sold the structure which was obtained by Wilkin and Franklin on X-ray diffraction technique So Watson and Crick, the important feature given by Watson and Crick are that the DNA molecule is basically consists of the two strands. And these two strands, as you can see in this structure, you can see that these two strands are anti-parallel to each other and these are complementary to each other. What does that mean? These two strands are anti-parallel to each other. That means this is, let us say, 5' end, then the other end would be 3'. Similarly, this would be the 5' end and this would be the 3' end. On the 5' end, on the 5' end of the DNA, there is always a free phosphoric group. There is always a free phosphoric group. So there is always a free phosphorus. So the basis is on the 5' end of the deoxyribose sugar, there is phosphoric acid which is free. So free PO4 2 negative is present on the 5' end. Whereas in 3' end, the hydroxyl group is free. Hydroxyl group is free. How these are free, we will talk in this later on. So if we talk about these strands, these two strands are basically made up of sugar and phosphate. So this strand here and this strand here, the backbone is basically made up of sugar and phosphate. So backbone is made up of sugar and phosphate. So if we talk about this particular structure, so this must be here some sugar, then there is phosphate, then there is sugar, then there is phosphate, then there is sugar. These sugar molecules are connected to nitrogenous base, that means purine or pyrimidine. So these sugar molecules are connected to nitrogenous base, and these nitrogenous base here are basically connected to each other. That means this guanine here is connected to the cytosine of the sugar of the other strand. That means basically these nitrogenous base projects inside. So the nitrogenous base here, projects towards inside position. The backbone is made up of sugar and phosphate. The nitrogenous base projects inside. These two strands are anti-parallel to each other. Already described. The width of the DNA is constant. Width is 2 nanometer or 20 angstrom. So if we talk about this width of the DNA, the width of the DNA is 20 angstrom. So the width of the DNA is 20 angstrom or 2 nanometer. If we talk about the turn of the DNA, in each turn of the DNA, the turn of the DNA is basically 3.4 nanometer, And in each turn of the DNA, each turn, one helical turn, this is called what we call one helical turn, and this one helical turn is what we call pitch of the helix. So this is called pitch of the helix. So the pitch of the helix is basically 3.4 nanometer or 34 angstrom. And in each turn of the helix, there are 10 base pairs. There are basically 10 base pairs. So in each turn of the helix, there are 10 base pair. That means you can say that between the two strands, between the two base pairs, there is a distance of 0.34 nanometer or 3.4 angstrom. Further, he said that that adenine is bonded to thymine by 2 hydrogen bond and guanine is bonded to cytosine by triple hydrogen bond. That means the basic force which is providing stability to this double-stranded helix molecule is hydrogen bonding. That means two strands are linked to each other. Two strands are linked to each other by hydrogen bonding. Adenine is bonded to thymine by double bond and guanine is bonded to the cytosine by triple hydrogen bond. If we talk about these two strands, the most of the structure of this strand is basically derived from Wilkins and Franklin structure given by them and the Chargaff's rule. But this is something special discovered or observed by Watson and Crick. Therefore, this whole thing is basically what we call hallmark of Watson and Crick. So this is what we call hallmark of Watson and Crick. Why it is known as hallmark? They did something different. If we talk about all these all things were known. Like the backbone is made up of sugar and phosphate. Nitrogenous base is projects inside. They are antiparallel to each other. Width is 2 nanometer. The molar concentration of adenine is equal to thymine and guanine is equal to cytosine. So these all things were already known before Watson and Crick gave the three-dimensional structure of the DNA. What was not known is these two strands are linked to each other by hydrogen bonding. So this is what we called hallmark of Watson and Crick. So this is the structure basically given by Watson and Crick. Thanks for watching the video. Please hit like and subscribe button. If you have any doubt, please don't forget to mention it in the comment section. See you in the next video.